Hello dear students, we are discussing our gastrointestinal physiology. In that we have completed the part 1, we have discussed about the structure and function of the GAT, then GAT movement and secretions. We have completed the part 2 also, wherein we discussed about the GAT hormones, the digestion and absorption process, and the dietary fibers and brain gut axis. Now we are moving on to the last part, which is a simple part, that is the structure and functions of liver and gallbladder, which we'll be discussing in this particular video. And in the next videos, we'll be discussing about the functional tests like gastric function test, pancreatic function test, and liver function test. So today's topic is liver and gallbladder functions. It's a very simple topic. So I'll give you a brief discussion of what is the structure of the liver and liver lobule, and followed by the functions of the gallbladder. So let's get into the topic. So first, coming to the physiological anatomy of the liver. When you consider the liver as a solid organ, it is the largest solid organ, and it weighs around 1.5 kg, which is around 22% of the body weight. So it is going to be around 2% of the total body weight. And the basic functional unit is called as liver lobule. So we will be seeing the structure of this liver lobule, which is depicted here. So this liver lobules, multiple liver lobules will together form the liver. So this is going to be a basic representation of a liver lobule. And there is high amount of blood flow because it has so much of metabolic activities. So the blood flow is going to be around 1350 to around 1500 ml, which is also huge amount of cardiac output is being given to the liver because of its metabolic process. And there is one important region based on which the surrounding which the hepatic liver lobule is constructed, and that is nothing but the central vein. So this centralmost part, as you can see here, this is called as a central vein. So this central vein, what it does is it first empties into the hepatic veins. So this vein will first empty into the hepatic veins, and further on that they will empty into the inferior vena cava. So this is their structure. And the liver also has good amount of lymphatic flow. So let's see this structure. The central vein is there. It is connected directly to the hepatic portal veins. Just now we have discussed this. And there is a structure which is very, very important. That is the space of Dizzy. So whatever any bacteria is first being acted upon by the liver, later on they have to be sent to the lymphatics. So that space of Dizzy, as you can see from here, they are connected to the lymphatics. So they are connected to this lymphatic system. That's why it is quoted as terminal lymphatic system. So this is one important area. And there are various amount of cells, important 3 different type of cells, which are present in the liver lobule. So the liver cell plate, which is nothing but the liver cell, which is going to do the primary function of the liver. And there is another additional cell, as you can see in this diagram, elongated cell, which is nothing but the Kupffer cell. This Kupffer cell is nothing but a macrophage of liver, and they are going to destroy all the bacteria that is being filtered out from the GAT. And there is the endothelial cells. So these are the 3 important cells. And liver is going to produce a lot of bile, and they will be emptied into the biliary canaliculi. So this green structure is the biliary canaliculi. So this green structure you can see here, this is nothing but the biliary canaliculi. And if you follow the tract, it is going to get emptied itself into the bile duct. And the liver has huge amount of blood supply, and it also has 2 different types of blood supply. One is hepatic artery, another one is the portal vein system. So this is the overall structure, as we can say, the functional anatomy or the physiological anatomy of the liver. And these are the cells and the other systems involved. So now let's get to the function of the liver. The liver has humongous, so many number of functions. It can act like a blood reservoir also. So whenever there is a huge preload to the heart, what will happen? Whenever there is a stasis, the heart is not functioning properly, there will be stasis, and this backflow can be stored in the liver. And the liver generally it can store up to 450 ml. That is the average amount of blood it can be stored. So the range goes around 500 ml to 1 liter of excess blood it can store. And the liver has one important property that is called as a regeneration of the liver. So when a part of liver was resected, even in experimental animals, they have seen that the liver has the capacity to regenerate if the certain environmental conditions and all the growth factors are available. And one predominant growth factor they have observed, that is secreted by the mesenchymal cells. The mesenchymal cells which are present in the liver. So suppose a small part of the liver is resected, what can happen is the remaining area will have the mesenchymal cells which can help to regenerate this region. And for that, one important factor is required, that is called as hepatocyte growth factor. Hepatocyte growth factor. This factor is very, very important. And there are other factors like tumor necrosis factor and interleukin 6. They are also involved in the liver regeneration. And they have observed that after a certain limit of the size, it automatically terminates itself. And the termination is done by the transforming growth factor beta. How it works, still now it has not been identified. This transforming growth factor beta gives the signal for the termination of further regeneration. And the hepatic macrophage system, I have already told you one specific cell, that is nothing but the Kupffer cell, which is going to be involved in the macrophage system. So these are some of the functions of the liver. Coming to its metabolic function, it is involved in carbohydrate, fat, and protein metabolism. So liver, it's a huge machinery and it is continuously working. That's why it consumes a lot of energy and so many mitochondria are present in the liver. It acts as a beautiful source of glycogen. So whenever anybody is having food, the glucose will be converted into glycogen and they will be stored in the liver. And as and when suppose the person is starving or he is fasting for some reasons, what the liver will do is it can reconvert this glycogen stores back to glucose. So that's why it is called as glucose buffer. It is going to buffer it very nicely. And it also can convert the galactose and fructose to glucose, and it can form something called gluconeogenesis. The generation of new glucose can be done. That is the gluconeogenesis. Coming to the fat metabolism, it can oxidize the fatty acids and release the energy from them. It can also synthesize cholesterol, phospholipids, and lipoproteins. So all of it can do. And it also can synthesize fats in the presence of food, proteins, and carbohydrates. So fat metabolism, it can deliver or derive the energy from the fatty acids, and it can also reform the fats from the proteins and carbohydrates. Next, coming to the protein metabolism, one most important functions of the liver is formation of urea for the removal of ammonia. If this ammonia is not removed, the metabolic product is not removed, what is going to happen is this ammonia can enter into the central nervous system and it can damage the brain. So it can lead on to something called as hepatic encephalopathy. So this is one dangerous thing which can happen. But if the functionality of the liver is there, it will convert this ammonia and remove it in the form of urea. And it forms the plasma proteins. So all the plasma proteins in the body, almost 90% of them, except for a few gamma globulins which are produced by the plasma cells. Apart from that, rest all is produced by the liver only. And it also causes deamination of the amino acids. So these are the metabolic functions of the liver. Now let's go on to the storage functions. It can store humongous amount of vitamin A, vitamin D, and vitamin B12. So the vitamin A, even if the person is not taking any vitamin A, the reserves from the liver can sustain it for 10 months, whereas D can sustain it for 3 to 4 months, and B12, it can sustain it for almost a year. And it also stores the iron, like it stores some of the vitamins, it also stores the iron. It has an important protein which is called as apoferritin. What does apoferritin do is, this apoferritin can combine reversibly with ferritin and can store it. Reversibly means it can store the ferritin and at the same time it can release the iron stores as and when it is required. And it also forms the blood coagulation factor. The factor 2, 7, 9, 10 is formed in the liver. For this, the essential vitamin is vitamin K, which is involved in the carboxylation of these clotting factors. So it is very, very important. And removal of other waste also, they will detoxify so many drugs like sulfonamides, penicillin, ampicillin, erythromycin. So all these drugs, whenever you give it to the patient, major detoxification is done by two organs. One is the kidney and liver. So this detoxification also is done by the liver. So if this detoxification doesn't happen, there is high chances that the patient develops any kind of hepatic encephalopathy and it leads on to the liver. That's why when a patient is having some kind of liver cirrhosis or liver failure, these processes are not able to do it, and this cannot be easily done by any other organ. And it also excretes or metabolizes some of the hormones like thyroxine, estrogen, cortisol, and aldosterone. And one more important function we have already discussed, that is the formation of bile. A detailed discussion of bile mechanism and synthesis and what is its important function we will discuss in hematology, because the most important topic, that is jaundice, we will discuss in hematology. So right now I am not going to the details. So it is involved in the formation of the bile. Now we will be discussing about this accessory organ which is situated very next to it, which will help to store this bile. So that is nothing but gallbladder. So that is called as the biliary system. The bile is formed by the liver, but the storage is done by the gallbladder. And the bile, generally it is in the ratio, it is composed of bile acids, which should be maximal, that is around 10, the phosphatidylcholine and cholesterol. Generally they are in the ratio of 10:3:1. But whenever this ratio changes, especially when the cholesterol level goes high, what is going to happen is it is going to form the gallstones. It is going to form stones, which is called as the gallstones. And gallbladder can concentrate the bile. That is another important function by reabsorbing a lot of water. So when you do a comparison between a hepatic bile and a gallbladder bile, the percentage of solid is just 2 to 4%. But by reabsorbing a lot of water, it is going to be concentrated in the gallbladder, which is its important function. It will concentrate up to 10 to 12% of the solids. And even if you see the bile acids in millimoles per liter, it is less in case of the hepatic bile, 10 to 20, whereas it is concentrated heavily in case of the gallbladder bile. Coming to the pH becomes more acidic in case of the gallbladder bile. So as you can clearly see here, the bile is being very well concentrated by the gallbladder. Even if the surgery of gallbladder is done, still the hepatic bile can directly flow into the duodenum, but this concentration advantage will be missed out. So that is the disadvantage when we do gallbladder surgeries and remove the polycystic gallbladder. Then coming to the regulation of biliary secretion, the biliary secretion regulates tasks as soon as there is a food in the mouth. So what is going to happen? There is a sphincter called a sphincter of Oddi. What is the sphincter of Oddi? The gallbladder has the ducts which will be emptying into the duodenal area, and there is one guarding area which is called as the sphincter of Oddi. This sphincter of Oddi will start to release as soon as the food is there in the oral cavity itself. So it is just going to prepare it so that it can easily digest the substances. Then it decreases the resistance of the sphincter of Oddi. And the fatty acids and amino acids, they are very, very strong stimulant for one important hormone that is called as the cholecystokinin. We have discussed in detail about cholecystokinin in the GA hormones. But a small note here, the main important function of CCK is to increase the secretions from the pancreas. But another secondary important function of it is contraction of gallbladder. And CCK is considered as one of the most powerful contractor of gallbladder, which is also called as cholagogue. The substances which contract gallbladder are called cholagogues, and it is the most potent cholagogue. And the vagus and secretin, what they are going to do? They are going to increase the bicarbonate levels in the gallbladder secretion. And the substances which increase the secretion of bile. So contraction of gallbladder, we just saw it is a cholagogue. Some of the substances, they can increase the secretion of bile, and they are called as cholecystic. And which is one important substance which increases secretion of bile itself is nothing but bile acids. The bile acid itself will help to increase the secretion of the bile. So this diagram shows a small brief thing about how the secretion is being modulated. So when the nutrients enter into the GAT system, they will produce a CCK. This CCK sends the afferents to the dorsal vagal complex, and the vagal efferents come back both to the gallbladder as well as to the sphincter of Oddi. So first they will contract the gallbladder with the help of acetylcholine, and they will also relax the sphincter of Oddi with the help of nitric oxide and vasoactive intestinal peptide. So these two hormones we have seen in the movements also, wherein this nitric and acid and VIP, they were causing the relaxation. So here also they will cause relaxation of the sphincter of Oddi. And another stimulant for the contraction of gallbladder is nothing but our CCK, which is the most potent cholagogue. So this is how the secretion is being regulated with the help of various other inputs from the vagus as well as from the CCK. Then coming to the applied aspects, I have told you, whenever the ratio between the bile acids, cholesterol and phospholipids, they are going to change. Especially the levels of cholesterol is going to rise. What they are going to do is, it will cause something called cholelithiasis, that is nothing but the gallstones. And this gallstone is very notorious, so it can obstruct the flow of bile and it can cause something called as obstructive jaundice. Jaundice, again I am repeating, we will just do a very detailed discussion in hematology. So what are the causes for this formation of gallstones? One is the bile stasis, and second is the supersaturation of bile with cholesterol. That is, the cholesterol levels are going to increase, or even the stasis. Whenever there is a stasis, even thrombus can form in blood in a similar way. This bile stasis can form stones here. And there are some factors called as nucleation factors, which is going to form which is going to help and form these kinds of stones. So this is all about the discussion about structure and functions of the liver and gallbladder in a brief discussion. And what is the surgery that can be done for this gallstones is nothing but cholecystectomy. So nowadays it is being performed laparoscopically. Previously it was an open surgery. So if there is not a complicated surgery, generally they prefer doing a laparoscopic type of surgery. So I hope it's clear. If you have any further doubts, kindly drop it in the comment section. I'll be happy to address your doubts. Thank you for watching the video. Stay tuned for more such videos. Thank you.